Welcome back to Dr. Liu's VCE Biology. Today's topic is DNA manipulation techniques and applications from the VCE 2019 biology exam. Let's learn together. If teachers, parents, or students see mistakes in my explanations, please give me your feedback. A transgenic organism has been genetically modified to contain a gene from another species. For example, a certain yeast can be modified to express a human gene. This yeast can be accurately described as a transgenic organism. A key benefit of using Bt corn, which expresses an insecticidal protein, is the resulting decrease in traditional insecticide use as Bt corn cultivation increases. This reduction helps lessen negative impacts on ecosystems. DNA ligase joins two DNA fragments by forming phosphodiester bonds between the two fragments. When restriction enzymes cut a plasmid at 3 restriction sites, they produce 3 DNA fragments. This demonstrates the principle that for a circular DNA molecule like a plasmid, the number of fragments generated equals the number of restriction sites cut. However, the standard answer is C. I don't think that it is correct. Using a blood test, individuals can be screened for 500 severe or deadly recessive gene mutations. Genetic screening is capable of detecting disorders like cystic fibrosis, PKU, albinism, and sickle cell anemia, among others. Since an individual's blood sample yields only a small amount of DNA, the polymerase chain reaction, PCR, is employed for amplification. The PCR process involves 3 key steps: denaturation at approximately 90 degrees Celsius, annealing to allow primers to attach, and extension where Taq polymerase copies the DNA strands. After amplification, the DNA is loaded into a well of an agarose gel. Several important factors affect the migration of DNA fragments through the agarose gel. These include the size of the molecules. Larger molecules move more slowly. The charge of the molecules, DNA's negative charge causes it to move towards the positive electrode. The length of time the voltage is applied, insufficient time, may not allow complete migration. And the concentration of the agarose, a denser gel resulting from higher concentrations, slows down molecular movement. Discovering that a couple planning a family, or already pregnant, both carry the same severe or deadly mutation raises major ethical and social implications. Ethical issues revolve around potential termination decisions for a fetus, the moral question of having a severely affected child, and accessing alternative reproductive technologies. Socially, implications include the societal cost of options like IVF, the psychological toll on carrier parents, and questions about the best use of public funds and required societal support. Accounting for 13 of the total 120 assessment marks, 10.8% of the assessment, the DNA manipulation techniques and applications section consists of 4 multiple-choice questions worth 4 marks, and 1 short answer question worth 9 marks. If you need my help, you can add me on WeChat or WhatsApp, or send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.